right, our next speaker is Kristina Kintziger, Claire M. Hubbard Professor of Health and Environment and the Associate Director of the Water, Climate, and Health Program at the University of Nebraska Medical Center. And she's going to talk about spatial patterns of PFAS in drinking water near point sources in Nebraska.
Don't know why that's stepping through the the names on here. So like the last speaker, I will also give a disclaimer. This presentation is the result of a former PhD student's dissertation who is now graduated, but she couldn't be here today, so I'm filling in for her, so I will try to do as good of a job as possible. So this is part of a larger project that we conducted looking at the presence, the types, the associations with health effects taken from rural water, drinking water samples around Nebraska, around known and potential point sources. In this study, the sampling was done between February and September of 2024, and we will focus on one part of that, the spatial patterns. So as a previous speaker discussed, there are a variety of sources for PFAS that are important for human exposure. For the purposes of this study, we were most interested in sources related to landfills as being potential point sources of PFAS, and then also military training sites around Nebraska that were most likely to be using the aqueous film forming foam AFFF at their sites. So those are the two primary pathways of interest. And then also, we are most interested in ingestion as the primary route of exposure. And that is probably, in terms of the general population, the most important route of exposure. Inhalation minimally contributes to total intake for humans, but it is still being investigated as a source specifically related to cosmetics and clothing. Skin contact is probably more important for occupational exposures, and then other routes include vertical transmission, maternal exposure to fetal transmission, or through breastfeeding. So this study just focused on one small part of the overall human health risk associated with PFAS. So we were again focused on PFAS exposure through drinking water, and we do acknowledge that there are other routes that are important that were not included. But we felt like this was a good start. So first of all, there are limited data available on PFAS in drinking water in Nebraska, specifically in private wells. The USGS has done sampling at one site. NWEE began testing select sites in 2020, but none of the large monitoring studies being conducted by US EPA, USGS, or CDC have included Nebraska sites primarily, so to date, and then also most of the existing sampling is around the public water system. So we wanted to focus on drinking water in private wells. So epidemiological studies have reported positive correlations between PFAS concentrations in water and PFAS concentrations in blood samples, specifically serum. And because this was part of a small pilot study, we didn't have the funds to conduct the biomonitoring, so we are using the drinking water concentration as a proxy for exposure. So again, just to review the health effects that have been associated with PFAS. On the left, we have those with the strongest links. So reproductive, cardiovascular, hepatic, metabolic, immunologic type outcomes. But then there are also some ones on the right that are associated with emerging evidence. So reduced cognitive, motor, and language development in infancy, behavioral issues in childhood, reduced fetal lung development, epithelial barrier function, risk of airway damage. A lot of this is from animal studies. We do lack the human evidence so far, but studies are ongoing. But in general, the mechanism for action for PFAS are poorly understood for a lot of these. So the overall objective of this study was to investigate the presence, concentrations, and patterns of PFAS in residential drinking water in rural Nebraska, to characterize the risk to human health and explore associations with health outcomes. So it is part of a larger study. I'm going to focus on aim 1, exploring the spatial variations of PFAS patterns relative to the sources of contamination in Nebraska. Our study setting, again, was residential communities near solid waste landfills and military sites in rural areas. For the water samples, we tested those using US EPA method 1633, which tests for 40 PFAS analytes. And a little bit more about the sampling. So what we wanted to do was focus on our known point sources, military sites. So there are 5 that were initially selected, but then we narrowed it down to 3. Yes, 3 that were training sites that are more likely to use the AFFF. And then we also wanted to look at landfills, which are a probable site, our source of PFAS, depending on a lot of other factors like depth of groundwater, things like that. But we used the US EPA ECHO database to identify landfills across Nebraska. We limited those 25 that resulted from that search to 4 based on certain criteria. For example, we wanted them to be in rural areas, geographically spread across the state, and also to have at least 2,000 households within a 5-kilometer radius of the landfill so that we could sample enough households to be meaningful. And again, we excluded sites both for military sites and landfills close to Omaha or Lincoln. And in partnership with the Nebraska Water Lab, Sarah developed a standard operating procedure for sample collection that included quality assurance, quality control, and method validation components. The samples were all taken from either the tap inside the home or from an outside hose bib if it was from the same source as the drinking water in the house. Not part of this presentation, but I will just share, she also did surveys with household members. So there was one part that was just about the household to get more information about water sources, usual patterns of consumption, and then an individual level survey for health outcomes for adults in the household who were willing to participate. For this particular aim, again, focusing on the geostatistical methods, we decided to include only the top 5 PFAS analytes or compounds in our spatial analysis and use the sum of these 5 as the measure of interest. Each site was analyzed as its own cluster except for Camp Ashland and the Meade training site because they were close enough in proximity that we needed to combine them. For the spatial patterns, we used empirical Bayesian kriging, and then for assessing spatial autocorrelation, hotspot analysis, including Moran's global eye and local Moran's eye. I should also say before I talk about the spatial autocorrelation, in her samples, PFOSA was the dominant compound identified, followed by PFOA. PFOS was low, so long-chain PFAS were the most frequently identified types in this study. So this slide shows you the results of the spatial autocorrelation. Because of sample size around the other sites, we were not able to do this analysis for all of the locations. And with the Meade-Ashland combined site, we did find areas of hot spots and cold spots. So basically, the global Moran's eye did indicate the presence of spatial autocorrelation, indicating geographic features are the primary factor. And then the local Moran's eye showed these hot and cold spots, indicating that this was not the result of random chance. So the hot spots were closer to the Camp Ashland military site, and the cold spots were closer to the Meade training site. There was also one low-value outlier, so it was an outlier like low values surrounded by higher hotspots, and that was located north of the hotspots. Then for the spatial patterns, there were 3 sites that overall had very low probability. So the outcome for these for these particular analyses were the probability of concentrations exceeding the EPA MCL of 4 nanograms per liter. So basically, these three sites here showed low probabilities overall for any of the samples to exceed that threshold. We did have some areas that showed higher probabilities. So while overall the Greenleaf training site in Hastings mostly had lower probability of exceeding that threshold, they did have one area in the Northwest that had maximum probability of exceeding the threshold between 48 and 80%. Then Plattsmouth had a very well-defined, that's the one on the right over there, had a very well-defined area of higher probability of the threshold to be exceeded, specifically in the west, to the west of the landfill. And then Camp Ashland and Meade training sites defined cluster around Camp Ashland, indicating higher probability of threshold to be exceeded. So in general, PFAS in drinking water is common in rural Nebraska, but at low concentrations, generally below the 4 nanogram per liter threshold. And the spatial patterns that we identified were generally diffused. There were some spatial patterns identified specifically at the two sites around Plattsmouth and Camp Ashland and Meade. And then hot and cold spots were identified at the Ashland-Meade combined site. So what this means overall for public health, this helps us to identify priority areas for further investigation for PFAS contamination and potentially help us in prioritizing mitigation strategies and areas that are in need of that. And then future directions include incorporating fate and transport factors impacting groundwater contaminants. I would like to acknowledge this was funded by DWFI and the Claire M. Hubbard Foundation. Thanking her other committee members and our collaborators at UNMC and the Nebraska Water Center and others. And we could not have done this without the help of all of the students who helped her collect these samples. So that was also an important component of this. Questions? Well, that was easy. Oh, okay.
Was there any factors that influence the decision using empirical-based spatial analysis as opposed to using a simpler method such as distance weighting to analyze spatial correlations?
Yes, we decided to use that method because it tends to perform better with smaller sample sizes. So that was primarily the decision. We ended up with 192 samples overall distributed across those 7 sites, and that was less than our target sample size. We just couldn't get a lot of people wanting to participate, so that is why we ended up using that method.
In Sarah's survey, she asked for any kind of like point of use treatment of both that impacted the results that you saw as you were testing.
She did ask that, and I do not remember the result. Like, I don't remember how that might have impacted, but she did ask that question. One of the questions that we probably should have asked that we didn't was like well depth, but maybe not everybody knows that information, but she did assess that using Nebraska Water Center has registered well depth information, and then the Nebraska Water Quality Clearinghouse also has depth to groundwater information, and there weren't any consistent patterns with that. So, but no, I do not remember the result of treatment.
Were they all registered well, or were they registered?
I don't know if she asked that either.